Hello everyone, I am Niels van Putte, and as already introduced, I did my master thesis about groundwater flow in freshwater tidal marshes. And the reason why I studied groundwater flow is because groundwater flow has big implications for the entire ecosystem function. For example, it determines for a large part the nutrient cycling, but also the vegetation development. And the reason why I made a comparison between a natural and a restored tidal marsh, I will explain with this animation. So tidal marshes are typically built up of sediments that were deposited by the tidal river over hundreds of years. But in the past centuries, many of these tidal marshes were embanked and used for agricultural purposes, the so-called polder areas. While the sedimentation kept going on in the natural marshes, the same is not true for the polder areas. Instead, the soil gets compacted by the use of heavy farming equipment, but also by the subsidence as a result of constant soil aeration. Now, more and more, these polder areas are transformed back into restored tidal marshes, and what happens then is that new vegetation develops and sedimentation occurs again, so that in the end, we end up with a situation where we have in the natural marsh a soil consisting of only the deposited sediment, and in the restored tidal marsh, a thin layer of newly deposited sediment and an underlying layer of the thick, compact polder soil. Now, because groundwater flow is inherently dependent on soil properties, we might just expect that there is a difference in groundwater flow between the natural and the restored tidal marsh. And that is what my master thesis was about. So I did my study in the freshwater part of the Schelde estuary, in the natural marsh, the Plaat, and the restored marsh, Lippenbroek. And in those two areas, I made a transect of monitoring wells approximately perpendicular to a tidal creek. And in those monitoring wells, I measured the water level, which resulted in these animations, which is a cross-section of the creek and the marsh platform, and the blue line represents the groundwater level. So what we see is that when the tide rises, the water quickly infiltrates in the soil, and when the tide then recedes, the water level falls back quite fast, about half a meter each tidal cycle. In the restored tidal marsh, we observe something completely different. Here, when the tide recedes, the water level only falls back a few centimeters, a little bit more close to the creek, but we clearly see that on average, the groundwater levels are much higher in the restored tidal marsh compared to the natural marsh. So then, to explain this, we took a look at the soil structure, and we did this with a very innovative technique called micro-CT scanning, which enabled us to look at the structure of the soil. We were mainly interested in macropores, the large empty spaces throughout the soil, here visualized in blue. And we can also stack these images together to get these 3D renderings of the soil. And then we saw that in the natural marsh, these macropores can be seen throughout the entire soil profile, while in the restored tidal marsh, we only encountered them in the upper layer, also a little bit in the lower layer, but around 100 times less. So then laboratory tests indicated that as a result of this, the underlying layer forms a barrier for groundwater flow in restored tidal marshes. So to summarize, what I found in my master thesis is that the former agricultural historical land use led to an altered soil structure, which then ultimately led to reduced groundwater flows. And now at this moment, I'm doing a PhD research where I will further investigate the effect on soil structure, but also how this reduced groundwater flow affects the nutrient cycling. And then also I will do some experiments to test which possible soil treatments or soil amendments can be used in future tidal marsh restoration projects to enhance the nutrient cycling and water quality improving function of future restored tidal marshes. Thank you for your attention.